Hello, this is Don Lee. I'm going to organize myself with this PowerPoint here. And I am an emeritus professor, recently retired from the University of Nebraska at Lincoln. My career was in the agronomy and horticulture department, and I spent most of my time teaching and taught entry-level courses in genetics and plant science. So here's a picture, a series of pictures that a former student took in a video project that she had to kind of show the approach that we take in learning in genetics, where we give the students the opportunity to work with us in small groups and work together. And this was the absolutely most fun I had in teaching genetics when I got to work with students in this way. So I'm going to share with you with this video the approach that I took when I gathered with students the first time we met in the lecture hall and got started on our genetics learning. And so here are the goals that I would have for the students when we gathered that day. First time together in the lecture hall, we have to do some ice breaking and promote peer learning. We're going to review a little bit of genetics they would have come in with some understanding of from their past biology learning, but the focus of our time together is to work like an applied geneticist, and you'll see what I mean by that. And we start kind of where genetics started historically with the work done by Mendel, but you're going to see I don't dwell on Mendel's research work. I bring in a different story that I think has some advantages. So this is what I'll share with you. And so I'm going to start then with the story that is really the kind of the center focus of our time together. So we can see here a little satellite shot of the world where I've been living, where I live in the forest of Lincoln, Nebraska, surrounded by the tall grass prairies that are now converted into farms. And I got a call one day when I was at work in the office. Former genetics student had a colleague from work who raised Labradors, and he had a really nice black female Labrador that he wanted to breed and have Labrador puppies and just sell those puppies on the market, find homes that would be motivated to purchase and adopt a Labrador. So the female, as I mentioned, was black, and the male that they identified to be the father of the litter was a yellow lab. And so the owner wondered if they were likely to get both black and yellow puppies. Could they predict the future? Was enough known about the genetic control of coat color so that we could make this kind of prediction, because it mattered to them. They knew they would have some advantages in marketing those puppies if they had some choice. They had both yellow and black puppies. Now, I'd spent a lot of time being a genetics teacher, and with curiosity and with teaching purpose, I'd learned a lot about what was known by dog geneticists' work about the genetic control of coat color in Labradors. And so I said, yeah, it is very possible to make some reasonable predictions, okay? And I was confident in that because coat color inheritance was a very important question that dog breeders had. They were funded to do research, so professionals were working with the breeders, professional geneticists were working with the breeders, and then they would share their discoveries because often they were funded by taxpayer dollars. And so I could go to the peer-reviewed research articles like this one and learn what the dog geneticists had learned. Now, I've selected this one because one of the co-authors, Edward Cargill, was a former genetics student of mine, and Edward was got his PhD in genetics at Texas A&M and was a canine geneticist. So in studies like this, Edward and his colleagues would share what they had learned. So I'm going to point out a few of the things that that they did. Let me see here. This is the abstract. I'm going to focus on just a few things. So first of all, they were hybridizers. They would cross Labradors oftentimes with other breeds in order to put themselves in position to better study the traits they were interested in. In this case, it was genes that would control coat colors. Okay, so there's the black coat, there's the yellow coat. They also had a third coat color in the families they worked with. But many of the ideas that I want to get my students confident in are ideas that geneticists like Edward and his team would use. So ideas like alleles and dominance and uh, and the idea that they would, I think I have the term here, that they would be looking at crosses that were segregating for those coat colors. That's terminology and conceptual understanding that's a goal of mine, and I'm trying to engage my students in that right away. So relating to the discoveries of the dog geneticists was something that I could do. And so in this particular story, our ability to predict the future uh, was not that difficult. I could tell from the mother's history that she was a hybrid. She had both yellow and black parents in her history, and therefore it was highly likely that the puppies from this particular cross would have both colors, both the black coat color and the yellow coat color. I could even take it a step or two further and predict the probabilities of each of the color types. And with Labradors, you usually get a litter, so the chances were pretty good. As it turned out, I kind of kept up with the story, and once the puppies were born, my wife and my daughter and one of my sons went out there, and I wanted to shoot a video of the family that I could use in my genetics teaching. And as it turned out, we became so enamored with the puppies that we adopted one of them, purchased one of them, Winston, and not just because of his yellow coat color, but because we knew Labradors had a lot of excellent genetically controlled characteristics or phenotypes that would appeal to us as dog owners. He got along great with the other animals in our family, humans included, and Winston's been a great choice for us to add to our family. So this was the story that I wanted to engage my students in. It was a good story that they could relate to, and it was a story that showcased the key inheritance biology that they probably came to our genetics class with. So I should take just a minute here and emphasize that there's a couple of different settings that we use to gather with our students. So one of the settings, once a week, we gather in a 2-hour recitation lab setting where the students work in groups and focus on problem solving. And in this setting, it's really a team sport. It's team learning by putting the students in groups, and also team teaching. I have undergraduate and graduate students that work with me as TAs, so the students get a lot of interactivity, a lot of hands-on work that goes into their ability to apply genetics concepts to solving genetics problems. And working with the students in this way is where the learning really happens. The students tell us that. We can see that because of the good vibe that we can generate in the lab. We also gather our students, like a lot of college classes, in large lecture halls. And here is 199 Plant Science Hall. It can fit 200 students, and so we half fill it typically with our genetics course. And in order to better promote peer teaching, I started bringing quizzes that the students would take, and they could work with each other on these quizzes. Here they're working on a quiz that they pull up in their learning management system, so they work on it on their computer, and this is at the end of the lecture. Now, this is the way I did it for many, many years, but more recently I changed my approach, and that's what I'm going to share with you how I did that. I changed my approach because I found a better way to promote this peer learning, and I could apply it to the goals we had no matter where we were in the flow of the genetics learning in the course, whether we were reviewing like we did today or in this first lecture, but we could get the students to work with the kind of thinking that geneticists had. So the key really to this change was to adopt these immediate feedback assessment techniques, these IFAT scratch quizzes. Okay, so I would hand these out to groups of students. They would kind of self-select into groups of 2 to 4 students and all work together on the same card. And if they knew the answer on their first choice and scratch, they'd see the star. If they didn't get a star on the first choice, they would say, well, we didn't get that one right. What else could be the right answer? And they would scratch until they discovered what the right answer was. So that instant feedback, that peer communication, and that iterative approach to learning really engaged them. And I always told my students the number one goal I had is to have us gather in lecture and have no one fall asleep. Okay, that seemed like a pretty low bar, but if you can keep your students alert for an hour or more because they're working together using their brains, that's success. Okay, so here are the rules that I used. And again, I use these scratch cards, and I'll provide you with the information that you could use to find the vendor for these cards today. Okay, so using these scratch cards, we could review genetics, and we could. So I'm going to share with you, some of the review. So I jumped to a number of the different conceptual areas that we tackle in our genetics course with this review. So they've probably studied chromosomes and their relationship to genes. So what we do is keep the context focused on dog biology. And it's amazing the amount of phenotypic variation among the different dog breeds, but all of these dogs have the same karyotype. They all have the same chromosomes, they all have the same genes on those chromosomes. They don't always have the same alleles, and that's where we get the variability from. So I can ask them questions about the relationship between genes and chromosomes, and I can add in new conceptual areas that they probably haven't had, but we'll get in genetics, like the use of molecular cytogenetics with fluorescent in situ hybridization. But the emphasis is that these techniques show us better the relationship between genes and chromosomes, but help us interpret the data that confirms the idea that when chromosomes replicate, the genes on those chromosomes replicate, and because the chromosomes are in pairs, the genes are in pairs. And that's really a key idea that we're going to work with in this course. Okay. So our learning goals are focused on dogs and focused on active learning, using these scratch quizzes to guide the students to work together. And I'm going to emphasize again that I'm using this dog biology as a better biology story. But it occurs to me that because the audience watching this video is probably educators like myself and not students, another opportunity you have with this story is to advance a student's understanding of how science works, how science advance works, how the discovery made by one scientist is really a part of a continuum of learning because scientists share their work with others. And Mendel's story is really excellent for that. Okay, so we're going to focus on the use of these Punnett square diagrams and to illustrate the genetics concept that I want the students to be able to apply that day. But most students know that we call these Punnett squares. We don't call them Mendel squares. So I've got this little hit pause here. I am going to pause this and go to the web. Now that the web is got so many robust resources. We are really set up to think about this idea of how does scientific discovery advance in the scientific community. All right, so I mentioned Punnett squares, and so you can get online and you can find one of the early publications by Punnett and a colleague of his, Bateson, that goes through the inheritance experiments of F1 and F2 generation that are really prototyped on the approach that Mendel used in his inheritance studies. But Punnett worked with chickens, with pig feather color in chickens, and if you take a look at the citations, they're citing Mendel and Mendel's ideas in this study. They're using things that we use today, pedigrees, in their paper, and they also use these square diagrams to show how the gametes that get made by the parents will randomly come together to form the offspring. And really, it's the work that Punnett shared with fellow scientists where we first see these square diagrams showing the idea that Mendel first proposed. So let's take a look at Mendel's paper. Let's see here. Here it is. Mendel's paper was published, decades before, and he was a hybridizer. He made crosses between different varieties of pea plants, and what he was really interested in is trying to understand what other hybridizers had observed. Okay, hybridizers would often be the folks that were working in plant breeding or animal breeding, trying to either maintain a particular variety, maintain the characteristics in a variety, or trying to establish varieties that had new combinations of traits. But what he had discovered, what the hybridizers had often seen, was that as they did this hybridizing work, they would see that offspring would revert back to one of the parents. The offsprings in some generations would have the characteristics that appeared to have disappeared in the early cross. And Mendel wanted to understand that phenomenon, so he went after it very systematically in this paper that he shared and published, but took a while to be rediscovered. Okay. I should also point out that now you can get online and in resources like Wikipedia, gain some history of the work of scientists like Mendel, and you can discover that the era at which they worked, the 1840s, and this is where it's going to connect to our dog genetic story, coincides with the era in which the Labrador dog breed was first developed by dog breeders. So the practical application of genetics in the development of breeds like the Labrador breed was happening at the same time that Mendel was making his discoveries using pea plants as a model organism. I'm going to go to a couple more resources. One of the things that you got to make sure you understand, though, if you're going to use peas, is an example is how pea reproductive biology works. And here's a really excellent picture I found. And when I go to the website, this is actually a very excellent website that was put together by this team of teachers, scientists, Doug and Jean, and they try to, this is a fabulous way to learn about Mendel and his work with this free resource. These are the characteristics that he approached. Here we see a diagram of the reproductive structures, but it takes a while for students to appreciate plant reproductive biology. And with the group of students that I serve in my genetics course, many of them have not really worked with plant biology, and this would be something that they would have to understand. And you know what's really cool about this site is not only the choice of learning targets, the visualization they provide, but also these little videos. Let's watch a little bit of it here.
Segregation that says that every individual possesses two alleles for any particular trait, like for example flower color, and which allele a parent gives its offspring is completely random. The offspring then has one allele from mom and one allele from dad, and of those two alleles, the dominant one is the one trait that the offspring will express. If and when both of the alleles happen to be recessive, only then will the recessive trait be expressed. But Mendel went even farther with his pea plants, and no, I'm not going to shut up about pea plants. It's fascinating, okay? And he got...
There you go. You know, he does a great job of ripping through the ideas that Mendel shared in this video, but he also is humorous about the idea that Mendel worked with peas, and most of you aren't going to be excited about peas. Okay, so therefore, I think it's important to bring in a biology story that your learners can relate to. I should also point out that I've developed, worked with colleagues to develop web resources, and one that I assign my students to read. They don't often read it, but it's available for them. Connects what plant breeders do with the principle that we're going to focus on in this first lesson. So I am going to go back to my PowerPoint here and emphasize that you know, I want the students to appreciate the biology that's embedded here in this story, and we're going to use the discoveries that Mendel used. But based on, you know, what I just shared with you, I'm going to give you a little quiz here. I'm going to throw out some terms that we would use in our teaching and ask which of these terms were terms that Mendel used in his 1866 publication. Okay, gene, allele, gamete, dominant, recessive, segregation, independent assortment, linkage. Okay, if we take a look at that first row, ratio. Take a look at that first row. Mendel in his paper talks about gametes. He talks about the pollen and the egg made in the reproductive structures of the pea plants. He uses dominant and recessive. He does not use the term gene and allele. He uses factors and characters instead. So other scientists following up on his work coined terms that we use today. How about in that second row? He does not deal with genes that are linked. It's an important concept but Mendel didn't encounter it. He does discover the principles of independent assortment and segregation, but he doesn't use those words, those terms to describe them. He does emphasize that the mathematical analysis that he conducts on his large numbers of offspring, the ratios that he looks with, reveal a pattern and a principle that he feels could be applied to all the 7 different characteristics and the combinations of those characteristics that he observed being inherited in his families. All right, so I don't take the time to linger on this kind of progression of science, that first day gathering, but we are going to use these key ideas of Mendel. It's just that there's so much biology embedded in this model that this biological model that we call the Punnett square, that the reproductive structures have the ability to make these specialized sex cells or gametes. The female makes eggs, the male makes pollen or sperm, and then they randomly come together when the offspring are formed. But again, my students don't often connect the seed as being the baby plant, the offspring. And so rather than encumber their understanding of the genetics because they don't understand the reproductive biology, I choose an animal example as we get started, okay? So they're much more intuitively tuned into the concept that females make eggs as their reproductive sex cells, and males make sperm. They make them in abundance, and then if the opportunity comes for the male gametes and the female gametes to come together, they can form the zygotes, which are the genotypes made at random. Those are the principles that the Punnett square illustrates. We want to make sure they understand how the Punnett square as a model connects to the actual biology that is happening. And I think it's a little easier to do with animal examples. So that's what I do. So then what we do is put them in their teams in position to work like an applied geneticist, and we're going to apply those principles discovered by Mendel. So again, I'm gonna hit pause here, and I'm gonna share with you a slide set that's that provides the data that I'll give them that allows them to work like geneticists. All right, now I'll provide you with this slide set. These are just pictures that I've pulled off the web. I'm not attributing to where the picture comes from, but these are just cute pictures of Labrador families when the pups are not very old, but you can easily score the coat color phenotype. And what I want to point out is that the nature of these pictures, sometimes the pictures are such that you can do a good job of counting every puppy and assigning its phenotype. But some of the pictures I have, the counting of the puppies is a little bit more challenging. So there's always an inherent challenge in gathering biological data. And one of the things that the students discover is that even though they're all looking at the same pictures, they don't always get the same numbers. But they gather them, we share with each other the data that we're collecting, and that sets us up to thinking about some of the inherent challenges in conducting an experiment. Okay, so let me go back to my other PowerPoint here. I'll pause this. All right, so the students are, after they've done the review and then after they've gathered the data from going through the slide set, I have some more questions that ask them to think like a geneticist. So for example, litter 2 is all black puppies, and given the methods that I share, should they use these puppies in the litter? We're supposed to look at families where the parents can produce both black and yellow. Here they're all black. It looks like the parents might be true breeding for black, but they get another, I show them another litter that does have yellows in it, and so we can combine those puppies from both crosses. So yes, they should include this data in their inheritance. Okay. I ask them a similar question about this family that has not just the black and the yellow, but the brown. And so thinking about the experimental methods is an important part of what we're going to do. And then we also want to be able to apply the model. Okay, so the genetic model that I'm encouraging them to use is the one that they had learned about before, the Punnett square, depicting the inheritance of variable alleles that control coat color in our families, but although they're collecting the same data on the same families, they don't all get the same numbers. Okay, so I want them to work like an applied geneticist in gathering the data, and then I want them to apply this model based on the discoveries of Mendel to analyze the results of their data gathering. They're working like a geneticist, okay? And so they can come up with the expected numbers based on their hypothesis. And again, I'll share with you this quiz and what I think are the correct answers if you would want to use this in your own teaching approach. And then we have some questions that connect this to the biology of producing sex cells, where they randomly come together to form the next generation, and we use a Punnett square to depict that random nature. And then we put the Punnett square to work, where I ask them, okay, we have a model. What would you predict is the genotype, or what's the probability that this black puppy here has the heterozygous genotype? So working like a geneticist, making the kinds of predictions and probability statements a geneticist had is something I want them to work with. And again, they often get these questions wrong, and that's a part of the mission. These are low stakes quizzes that we give in lecture, way more points than the maximum they can earn. So they're learning by making mistakes. It's that I have to put them in position to do the thinking with the situation or the story that we're working on and the questions that guide their thinking. All right, so the principle of segregation gets us started, but the rest of the learning in that first week will include independent assortment. So I bring in a second trait, a gene that controls this exercise-induced collapse that we see in some breeds of dogs. So we can make probability statements there, but in this case, we're not just using the principle of segregation, we're also using the principle of independent assortment, and we can apply that principle to the inheritance of these phenotypic traits, including the inheritance of the sex chromosomes that control the male or female gender phenotype. Okay, so the last thing I'll do is give them a little preview to where the course will be going once we tackle molecular genetics, and I'll also give them a preview to a strategy that I use that will revisit this coat color trait when we learn about how genes work at the molecular level. If we can understand how the trait is passed on, how the trait, the gene controlling that trait was discovered on a certain chromosomal position, and then how the sequence of the nucleotides in that gene control its expression and the information delivered that controls phenotype, that really encompasses the goals that we have in this course. Okay, so as you can see, I cover a lot of ground, but I engage the students to work together to see how their collective thinking can correctly answer questions. It's okay if they can't. The key is to get started learning by applying genetics and making mistakes, but learning from those mistakes. Okay, so this is the goals that I have for the students in the first day. I'll share this and the supporting documents with you in this with this YouTube video.